So the next speaker now is Tina Singh from Grant Brown's lab at the University of Toronto.
Okay, great. Thank you for the introduction, and thanks to the organizers for giving me the opportunity to tell you about our work. I'm looking at how the RISC complex functions to maintain ploidy in budding yeast. So in the Brown lab, we're generally interested in studying genes involved in genome maintenance, which are essential for ensuring that the integrity of the genome is maintained through the cell cycle. So one source of genome instability that isn't commonly talked about are changes in ploidy. That is, changes in the number of sets of chromosomes that an organism has. Now, this may not seem like an apparent source of genome instability because many organisms tolerate changes in ploidy just fine. As an example of this, I'm showing you this perennial ryegrass species, where you can see in this photo on the left is a diploid wild-type plant grown beside its tetraploidized counterpart. And what you'll notice is not only are the tetraploid plants growing very robustly, they're actually much larger than the diploid plant. And so this is beneficial for farmers because you get more biomass per seed. But also it turns out that cows prefer the juicier tetraploid plant and will naturally eat more of it. So in this context, changes in ploidy are tolerated and are also beneficial. Of course, this isn't always the case. So in humans, unscheduled changes in ploidy can be very detrimental. And so here I'm showing you two karyotypes, a normal versus a breast cancer karyotype. And you can see in this breast cancer karyotype, every chromosome is apparent in almost 4 copies, and it's thought that this tetraploidization was a precursor to all of the aneuploidy that you see. Okay, so this is why we're interested in studying ploidy maintenance. And so several years ago, the Boone and Nizlo lab ran the entire deletion collection through the flow cytometer as a quality control measure. And they actually found a subset of 178 genes that, when deleted, caused spontaneous diploidization. And among the subset were several deletions of subunits of a complex called the RISC complex. And it turns out that 6 out of 7 of the non-essential subunits are critical for ploidy maintenance. And so we wanted to first confirm these results. And so we did a time course where we took a stable heterozygote of each of the deletion mutants, sporulated and dissected them to obtain a fresh haploid, and then we monitored the ploidy over the course of 100 generations with flow cytometry. And so first I'll show you this 102 deletion, and so this is the only subunit that, when deleted, does not cause diploidization, so it behaves like wild type. And you can see for the duration of this experiment, the strain remains haploid. Now, in a RISC2 deletion, you can see that after germination, the population is haploid, but there's an emergence of a 4C peak after 30 generations of growth, and the entire population is diploidized by about 60 generations. Finally, HTL1 deletion causes very rapid diploidization, so you can see after germination, the entire population seems to be diploid. And so this led us to our hypothesis that the RISC complex is important for ploidy maintenance, but the contribution of each individual subunit seems to be unequivalent. Okay, so what is the RISC complex? Well, first it stands for remodels the structure of chromatin. It's an ATP-dependent chromatin remodeler that moves histones around the entire genome. It has many roles in transcription, and it's also directly involved in double-strand break repair, cohesion, kinetochore attachment, and spindle checkpoint. Okay, so now that we identified this spontaneous diploidization phenotype, we decided to have a look at the morphology. And to do this, we collaborated with Yoshi Oya's lab. In particular, I was working with his postdoc, Shinsuke. And so in their lab, they have developed this software called CalMorph, which can measure hundreds of parameters of yeast. And so in this principal component analysis, we had Shinsuke compare the morphology of the entire deletion collection in blue with 100 replicates of a haploid wild-type strain in pink and 100 replicates of a diploid strain in orange, and then you can see the RISC are in yellow. So as we'd expect, the RTT102 deletion strain that does not diploidize clusters with the wild-type haploid cells, and so this strain is morphologically similar to a wild-type haploid. However, what was interesting is that the diploidized RISC strains actually are morphologically distinct from the wild-type diploids in orange. And so in data that I don't have time to show you today, what we found is that the change in morphology is actually due to the nature of how these cells diploidize. And so whereas a wild-type diploid is a MAT-A alpha, it turns out that the RSC diploids have diploidized through a non-disjunction event, and so are MAT-AAs. And so we're currently working on understanding how the relationship between non-disjunction and morphology. But this analysis pointed us to chromosome segregation, and so we wanted to learn how the RSC complex was facilitating proper segregation. And so to do this, we focused in on the HTL1 allele. So I'll remind you that the HTL1 deletion causes the fastest diploidization. And so we made these conditional alleles where we integrated a galactose-inducible HTO1 allele at HO. In a wild-type control strain, you can see that regardless of whether you grow on galactose or glucose, the strain remains haploid. However, in the HTL1 delete, overexpression will rescue the diploidization, but you can induce spontaneous diploidization by growth on glucose. So after 10 generations, we see the emergence of a 4C peak, and by 20 generations, the entire population has diploidized. So we used these conditional alleles to make movies, and so we decided to mark the spindle pole body, which is the microtubule organizing center, with green. So we marked the core component SBC42. And we also marked the histone H2A with red to denote the DNA. And so each movie represents an 8-hour time course where I've taken images every 30 minutes. And so I'm going to draw your attention first to the normal cell, just to orient you. The easiest way to watch these movies is to watch a single cell. So I'll draw your attention to the one in the middle. And when I start this movie, you'll see that this cell will bud. And you see spindle pole body duplication and segregation. And again, if you look at this cell in the middle here, you'll see budding, duplication of the spindle pole body, and segregation over and over again very robustly. Now, what was interesting is that when we depleted HTL1, we actually found 3 different categories of spindle pole body defects. So first, I'll show you this duplication failure. So you can see that there's a large budded cell that's failed to duplicate its spindle pole body, and so it's sort of just tracking around with its DNA. And what's interesting is that what eventually will happen is the cell will rebud right here. And so what we think is happening is that this daughter, which received no DNA, will bud off and die. The mother diploidized and then resumed the cell cycle and continued through the cell cycle. And so these 2 cells are now diploidized. Okay, the next category is an attachment failure. So I'll start the movie. There's a cell that's going to fly into the center here. And you can see that the spindle pole body has duplicated, but one spindle pole body is associated with the DNA, while the other one is sort of just tracking around in the daughter and not attaching to the DNA at all. And so again, what we think is happening is this daughter will bud off and die, and this mother has now diploidized. Finally, we see the third category called the separation failure. And again, this cell in the center here, it undergoes spindle pole body duplication, but for the remainder of this movie, there is no segregation. And so again, what we think is happening is this daughter will die because it does not receive DNA, and this mother has now diploidized with 2 spindle pole bodies. And indeed, we have seen movies where a single G1 diploid contains 2 spindle pole bodies, and it continues through the cell cycle, and it actually duplicates its 2 spindle pole bodies to 4 and will still segregate its DNA. Okay, so 2 takeaways from these movies. First, you'll notice that in all 3 cases of segregation defects, we see cell cycle arrest. So we think that these cells are still undergoing the spindle assembly checkpoint. However, the cells will eventually adapt and continue through the cell cycle and continue to propagate as a diploid. And the second thing is that it's sort of curious as to why the depletion of a single protein would cause so many different defects in spindle pole body function. So to try to understand the mechanism of how the RSC complex is influencing spindle pole body duplication, we got a hint when we did a synthetic dosage lethality screen. And so in this assay, I have the HTL1 shutoff strain, and I've expressed a high copy number plasmid containing each of the indicated spindle pole body components listed. So in growth and galactose, we see that all of the strains grow similarly. However, in glucose, we see a strong synthetic dosage lethality with 2 proteins, MPS3 and NBP1. And when we look at the corresponding flow cytometry, we actually see that, maybe best seen at this 10-generation time point, that overexpression of these 2 proteins actually causes more rapid diploidization. And so what's known about MPS3 and NDC1 is that they're very important for spindle body insertion. And so we thought we'd look at whether insertion was being affected in the HTL1 depletion strain. So we borrowed this assay from Sue Jasperson's lab. And so this is just a co-localization assay. So here we have the core component SBC42 now marked in red, and a nuclear spindle pole body component called SBC110 marked with green. So I'll walk you through each of the phenotypes. So starting with normal at the bottom here, in a large budded cell, you should have 2 co-localized spindle pole bodies. If duplication has failed, you would see only 1 co-localized spindle pole body. And here's the insertion defect, where you have 1 normal red and green co-localized spindle pole body, and then you see this red dot by itself. And so this signifies SBC42, which binds with itself to make a large duplication plaque. If it does not insert into the nuclear envelope, then SBC110 cannot bind to it on the nuclear side, and so this is why you see red by itself. And finally, we have this other category where we sort of just throw anything that has multiple spindle pole bodies into. So here's the data. In the wild-type case, we deplete HTL1 over the course of 8 hours, and you can see that all of the large budded cells are normal. However, in the HTL1 deletion case, we see that over time, first we see a delay in spindle pole body duplication, and then we see the emergence of this insertion defect. And so now we hypothesize that risk is important for spindle pole body insertion, but again, how is it affecting spindle pole body insertion? Well, we got a hint from this spot assay. This is the same spot assay that I just showed you. And it turns out that overexpression of this protein called NBP1 actually causes this strain to grow better, even better than the vector control. And it turns out that overexpression of NBP1 actually rescues the diploidization phenotype. And so what's known about NBP1 is that it needs to be imported into the nucleus through the nuclear pore complex, where it localizes to the spindle pole body to facilitate proper insertion into the nuclear envelope. And what we also found was that in 2 studies by the Wozniak and Wendt lab, they showed that the RISC complex actually binds to the nuclear pore complex in order to stabilize it and facilitate appropriate localization. So I had a look at nuclear pore complexes in the HTL1 shutoff strain, and so I used 2 markers, NIC96 and NUP170, marked with GFP. You can see that growth in galactose shows nuclear pore complexes that are healthy and diffuse around the nuclear envelope. However, when we deplete the cells of HTL1 by growth in glucose for 24 hours, we start to see aggregation of the nuclear pore complexes, which signifies stress. Okay, so if risk is affecting nuclear pore complex stability, and this is affecting NBP1 transport into the nucleus, we hypothesize that if we decrease NBP1 transport by a different mechanism, so by deleting its carrioferin, CAP123, we should also see the diploidization phenotype. And indeed we do. So this experiment was done by another graduate student, Minnie Hung, and you can see that the wild-type strain is haploid, while the CAP123 delete diploidizes over time and is completely diploid by about 40 generations after germination. So this leads me to our model, where the RISC complex binds the nuclear pore complex to facilitate the appropriate transport of NBP1 into the nucleus, where it localizes rapidly to duplicating spindle pole bodies to facilitate insertion and normal segregation. In the RISC deletion strain, we see that the nuclear pore complexes are stressed, and this decreases the NBP1 transport into the nucleus. And this results in failed insertion, and this affects spindle pole body maturation, as well as microtubule nucleation on the nuclear and cytoplasmic side. And we think that this is why we see so many different defects in the movies that I showed you. So with that, I'd like to thank my lab, my supervisor, Grant, who's here, Minnie Hung, who helps me a lot with this project, as well as our collaborators from the University of Toronto and Tokyo. I'll thank our funding source and all of you for your attention. I'd be happy to take questions. Thanks.
Once they've diploidized, the RISC mutants suddenly free of problems, or do they still have problems that make them sometimes tetra? diploidized?
Right. So if we look at the flow cytometry, we don't detect tetraploids. But what we can do is take an AA and an alpha-alpha from the same risk deletion background and mate them. So they form normal zygotes and become tetraploid, but we found that over time they were very unstable. They have decreased viability. And it turns out if you look at them under the microscope, if you look at the spindle pole bodies, they're enriched for having multiple spindle pole bodies. So what we think is happening is that they're becoming aneuploid and dying.
I'm interested in the return to cell cycle. Is this a typical adaptation response that you're seeing?
I think it is. I mean, they arrest for a similar amount of time in the adaptation studies and then resume the cell cycle. So we haven't really looked into it. Thanks.
So do you ever see aneuploidy showing up after you form the diploids? Because if you have multiple spindle pole bodies, then you would expect that they would become aneuploid also.
Right. So we usually just look at the strains over the course of 100 generations. It sort of depends. Sometimes we see some aneuploidy coming up, but they're relatively stable. But I haven't really looked past 100 generations. I'd suspect that the ones that are accumulating multiple spindle pole bodies would become aneuploid. Although we do see in our movies when there are 4 spindle pole bodies in a cell, sometimes it does look like segregation is occurring relatively normally. So.
And then multiple spindle pole bodies, do you think it's actually one nucleus that's big and has 2 sets of spindle pole bodies within one nuclear membrane,
Right.
Or do you think it's actually 2 nuclei next to each other?
Right, so I see both instances. So we have the nuclear pore complex marked as well, and so one question that we always ask is there a bit of nuclear envelope that's at least associated with the spindle pole body? And I would say in the extreme cases where I see up to 9 spindle pole bodies in a single cell, that's not the case, but when it's sort of on the lower end, usually it's associated with the nucleus, and sometimes it's one nucleus, and sometimes it's a few of them.
Okay, interesting, thanks.
Nice story, Tina. So following up on Bob Reed's question about adaptation, have you looked at RDH54 to see if the cells change their behavior? Because RDH54 protein is actually at the kinetochore as well and is important in adaptation. Probably haven't done it yet because you wouldn't have thought about it, but
Yeah, we haven't, but
it'd be a valuable experiment.
Okay, thank you.
Maybe you can move on to questions right
We-
now.
okay.
Can I catch you after?
Great, thank you.
Thank